Today's practical title is Petiole preparation of a Dicot plant. And this is a Dicot plant. And the name is Talinum triangulare or water leaf. Now, what's a petiole? A petiole is the stalk that supports the leaf blade and connects the leaf to the stem. So this is a petiole. Connect the leaf to the stem. Now, aim of the experiment is to prepare a cross section of a mature petiole of a dicot plant. So the materials I'll be using, we have distilled water in petri dish and sodium hypochlorite petri dish. We have different grades of ethanol. We have a safranin stain. We have a glycerol. We have a nail varnish. We have a slice and cover slip. A slice and cover slip. And a cover slide. Now the procedure. Now the freehand method will be applied to the preparation of this cross section by using a blade. And then the blade will be used to cut the section of the petiole after the petiole has been stuck inside a sterofil material. Then the section will be cut and placed inside distilled water to hydrate the cell and then transfer into sodium hypochlorite to decolorize the cell. And then later transfer into distilled water to remove the effect of the decolorizer. After which it will be placed on a glass slide, and then it will be stained with different grades of ethanol. And the purpose of staining with ethanol is to dehydrate the cell. Then it will be stained with safranin, which is a choice stain, so that the cellular organisms can absorb the stain. Then then a drop of safranin will be added to it for easy visibility under the microscope. After the staining, then the prepared slide will be covered gently with cover slip on one side to avoid trapping air bubbles. Then this excess stain will be wiped off, and then the nail hardener or nail cutters will be used to rinse the cover slip, or better say, to seal off the edge of the cover slip to prevent the addition of our stain. Then after the staining is done, the prepared slide will be examined under the microscope at times 4 objective, times 10 objective, and times 40 objective. So the staining proper. Now the petiole has been inserted inside the styrofoam material, this styrofoam material, and then a thin section is made inside the still water. Several sections will have to be done so that you get a very thin section. You have to cut several sections. Insert your petiole inside the material. This to make a kind of tissue, a kind of block of a styrofoam material, so that when you cut the section, you have a very thin section. This we're doing is we are trying to mimic a use of a microtome machine, which make a very good section. So you cut tiny section gently into the still water. You cut several section of the petiole. Now you cut into distilled water to hydrate the cells. To hydrate the cells. After which the cut section is transferred into petri dish containing sodium hypochlorite in order to decolorize the cells. So that when we are staining with our choice stain, it will pick the stain. This organism will pick the stain better. We leave inside the sodium hypochlorite for about 10 to 20 seconds for the sodium hypochlorite to decolorize the cell. After this, we transfer the cut section into another distilled water to remove the effect of that sodium hypochlorite or jig. We need to remove it from so that our staining will go smoothly. After you put inside the still water to remove the effect of the sodium hypochlorite, about 5 to 10 seconds, then you remove the cut section and you mount on the glass slide. The distilled water has hydrates the cells and also remove the excess effect of the sodium hypochlorite. Then the next thing is we have to dehydrate our cells gradually using different grades of ethanol. First, we add 1 to 2 drops of 50% ethanol and leave for like 5 seconds. You drain off and then you put 70% ethanol. Now we are adding different grades of ethanol so that we will not break the cells. And ethanol helps our cut section to be adding. So ethanol, the purpose of ethanol is to add in the cells. So we add 100% and then we drain the excess, we drain it off. So after we have added our cells, it is ready for staining. So we put 1 to 2 drops of safranin O stain, 1 to 2 drops of safranin O stain, and allow to stay for about 30 to 60 seconds. Leave about 30 to 60 seconds. After 60 seconds, you add, of adding safranin stain, you put 1 to 2 drops of glycerol. The purpose of glycerol is for easy visibility of our prepared slide under the microscope. After you have added 1 to 2 drops of glycerol, you gently cover the slide with cover slip from one end in order to prevent trapping of air bubbles. So you put cover slip gently from one side so that we can contain all the stain in a particular inside you can use the stain to contain all our stain sample. After this, after you have added glycerin, you clean the excess stain You drain off the excess thing, and then you seal the edge of the cover slip with nail hardener or nail varnish. Now we see the edge of this repair slide, edge of this cover slip, so that our slide prepared will not be dehydrated. After which we examine under the microscope at times 4 objective, times 10 objective, and times 100 objective. Thank you. So this is the microscopic view of the petiole preparation of a Dicot plant, as seen under times 10 objectives of the microscope. So above is the note for the video for you to check later. So if you find this video very useful, don't forget to like and share. Also, kindly comment if you have any question or addition, so we can get back to you in the next video. Thank you.